When I say cryotherapy, most people think of cryogenics, a science fiction method of hibernation made popular by Woody Allen's Sleeper, Tom Cruise's Vanilla Sky, and even Mel Gibson's Forever Young. But by definition, cryotherapy is any method that uses cold temperatures to treat disease. So cryotherapy can be as simple as sticking your foot in a bucket of ice water, or as sophisticated as using a surgical cooling device. And it's been around for a while. Ever since the days of Hippocrates, people have been using cryotherapy to reduce swelling, pain, inflammation, muscle spasms, and bleeding. However, there is no standard cryotherapy protocol. Some experts advise days of continuous cooling. Others suggest short cooling durations, with plenty of breaks. Our laboratory investigates surgical cooling devices and ways of making them safer. These devices circulate ice water through detachable pads and maintain localized cooling for long periods of time. But when used for extended periods, these devices are associated with non-freezing cold injuries, including tissue death and nerve injury. It's not completely clear why this occurs, but it's likely the result of reduced blood flow, or ischemia. Here you can see cryotherapy induces ischemia, reducing oxygen and cell nutrients and allowing for the buildup of toxic metabolic byproducts. In our lab, we've performed over 100 human subject experiments. We've tested multiple body parts, including knees, shins, and ankles. Each experiment begins with 30 minutes of baseline recording, followed by 1 to 80 minutes of ice water flow, and 30 to 240 minutes of passive rewarming. To conduct our experiments, we use thermocouples to measure temperature, Doppler probes to measure superficial blood perfusion, heat flux gauges to measure heat flow rates, a data acquisition interface, and NI LabVIEW Signal Express on our computers. Let's look at a graph of temperature versus time, divided into baseline, cooling, and rewarming. As expected, the temperature remains constant during the baseline period, falls rapidly during cooling, and increases again during rewarming. Perfusion, on the other hand, looks very similar initially, but it remains depressed long after skin temperature has returned to near baseline. It is our hypothesis that intermittent stimulation of blood flow should obviate ischemia as a source of injury. We've developed multiple devices and methods, filed patents accordingly, and are now developing a new generation of safer cryotherapy systems.